Activity-dependent plasticity is a change in the strength of synaptic transmission that is caused by previous synaptic activity. Activity-dependent synaptic plasticity can relate to changes in the presynaptic neuron, for example, a change in the amount of neurotransmitter released in response to each presynaptic action potential. Activity-dependent synaptic plasticity can also relate to changes in the postsynaptic neuron. For example, a change in the number of neurotransmitter receptors on the membrane of the postsynaptic cell.
Changes in the strength of synaptic transmission, or plasticity, can occur on both sides of the synapse, either by manipulating the probability of presynaptic release or the responsiveness of postsynaptic receptors. For instance, the calcium-dependent nature of neurotransmitter release leads to short-term plasticity in synaptic transmission. The accumulation of calcium following multiple spikes can lead to more effective vesicle fusion and thus facilitate neurotransmitter release, and ultimately the activation of postsynaptic receptors. On the other hand, the reliance on readily releasable pools of vesicles for neurotransmission means that as activity levels persist and vesicles are depleted, neurotransmitter release is depressed. While these are examples of short-term plasticity on the presynaptic side, there are also examples of longer-term plasticity on the postsynaptic side. Strong electrical stimulation of presynaptic axon terminals from one input leads to long-term potentiation of synaptic transmission, measured as an increase in the amplitude of the excitatory postsynaptic potential, or EPSP, connecting the two neurons. Critically, long-term potentiation, or LTP, is synapse-specific and limited to the inputs that underwent stimulation, perhaps mimicking formation of a specific memory. However, synapses do not just get stronger. Interestingly, weak electrical stimulation has exactly the opposite effect, leading to long-term depression of synaptic transmission, known as LTD. Again, the changes are synapse-specific, perhaps mimicking the selective loss of information. Calcium entry through the ionotropic NMDA-type glutamate receptor is linked to the postsynaptic changes underlying LTP. Activation of PKC phosphorylates ionotropic AMPA-type glutamate receptors, leading to a greater conductance and larger EPSPs. In addition, activation of CAMK2 mobilizes new AMPA receptors from local vesicular organelles, which also increase EPSP amplitude. Differences in the levels of NMDA activation have also been linked to LTD. During strong stimulation, higher levels of NMDA activation lead to greater calcium entry, promoting activation of protein kinases that trigger LTP, as above. However, during weaker stimulation, lower levels of NMDA activation lead to less calcium entry, promoting the activation of protein phosphatases that trigger the opposite response in LTD.
In introductory classes, you learned about activity-dependent synaptic plasticity, but neuroscience is an interdisciplinary field, so there are many words for this term. For example, you may also hear about neuroplasticity or just plasticity, and these terms relate more broadly to any change in neural organization. Activity-dependent synaptic plasticity is an umbrella term that refers specifically to changes at a synapse, and these include increases in synaptic strength, such as facilitation, augmentation, and potentiation, as well as decreases in synaptic strength, such as depression.
Activity-dependent synaptic plasticity plays an important role in learning and memory. For example, did you know that some types of learning require activity-dependent synaptic plasticity in specific parts of the brain? Fear conditioning, which requires activity-dependent synaptic plasticity in the amygdala, is a process that occurs when a neutral stimulus, like a sound or a light, is repeatedly presented at the same time as a naturally avoided stimulus, like a painful shock. Eventually, a test subject learns to fear the previously neutral stimulus. Experiments in rats have shown fear conditioning requires long-term potentiation, which is abbreviated LTP, in the amygdala. Recall that LTP is a form of activity-dependent synaptic plasticity that involves increasing the strength of synaptic transmission by inserting AMPA receptors into the membrane of the postsynaptic neuron. When this process is selectively blocked in the amygdala, fear conditioning does not occur, and a rat fails to fear a neutral stimulus even after that stimulus is repeatedly paired with a painful experience.
Okay, now that you've had a bit of review of activity-dependent synaptic plasticity, let's see if you can follow along with this example of NMDA receptor-dependent long-term potentiation, which gets abbreviated LTP. In this scenario, what we'll start off with is a glutamatergic synapse, meaning the presynaptic neuron is signaling with glutamate as its neurotransmitter. And what we'll see on the postsynaptic neuron is two different kinds of glutamate receptor. There are the AMPA receptors here, and you will recall that these are excitatory receptors. And then we also have the NMDA receptor. This is also an excitatory ionotropic receptor. But importantly, this NMDA receptor is permeable to calcium and also blocked by magnesium ions when the voltage is low. Okay, so what we might find at this synapse is if you have a lot of activity at this synapse, a lot of glutamate is exocytosed. That glutamate will bind the AMPA receptors, and via these AMPA receptors, there will be an excitatory current that depolarizes the postsynaptic cell. As the voltage in this postsynaptic membrane, or the postsynaptic cell, increases, the glutamate, which is also binding the NMDA receptor, causes it to open. And because the voltage is elevated, the NMDA receptor is not blocked by magnesium ions. So there will be further depolarization via the NMDA receptors and also an influx of calcium ions. This influx of calcium ions is important because calcium ions will activate intracellular protein kinases. And these protein kinases will act on a reserve pool of AMPA receptors, causing them to be inserted into the membrane. The kinases will also phosphorylate the existing AMPA receptors. And so the next time we get activity at this synapse, it will be stronger for two different reasons. One, the existing AMPA receptors are phosphorylated, which allows them to pass more current. And also, additional AMPA receptors have been inserted into the membrane. So the next time glutamate is exocytosed, it will cause additional increase in voltage by activating a larger number of AMPA receptors.